Cool. We'll get started now. So thank you everyone for joining this month's Funk seminar. I just got a few announcements on behalf of the Biogeography Society. Our biannual conference website is now up and abstract submission is now open. So you have until September 1st to get that in if you want to secure a talk. And then standard registration will begin in November. If you have any questions about the conference, all this can be found on our website, biogeography.org. Um, at the end of the talk, we'll have a quick Q&A session. So if you have a question, you can either type it in and we'll read it out loud, or you can raise your hand and we'll call on you and you can ask your question directly. And yeah, thank you again for attending this month's seminar. All our talks are recorded and then uploaded to YouTube. So if you want to rewatch this talk later, you can. And then for next month, we will have Mario Mora. Yeah, and then I will pass it to Mario to introduce our speaker.
Thanks so much, Raymond. And thank you all for coming. Just to follow up on Raymond's slides, if you want to attend the Biogeography Society meeting in Denmark, please make sure to register as soon as possible, or at least as soon as you know the outcome of your abstract, because there actually will be a limit in terms of the amount of, or the number of participants we can admit this year. And that's because the venue is a little smaller and can only, absorb so many people. So if you do want to come, and I hope you do, please make sure to follow those deadlines and get your spot early. But today, it is my absolute honor to, in a second, introduce Dr. Jamie Kass as this month's speaker for our Funk Lecture Series. My name is Marta Jarzyna, and I'm the Vice President for Public Affairs and Communications at the International Biogeography Society, for those of you who don't know me, maybe. Dr. Kass is an associate professor at the Graduate School of Life Sciences at Tohoku University in Japan, where he runs the macroecology lab. Dr. Kass obtained his BS in biology and BA in English from State University of New York in Binghamton. He then went on to get a master's in environmental management from Duke, and then a PhD in biology from City University of New York. So I see that biology won over English, which is a score for our field. He then became a JSPS postdoctoral fellow at the Okinawa Institute of Science and Technology Graduate University before landing a professor position at his current institution. Dr. Kass's group uses species distribution models and other analytical tools to investigate how global change affects biodiversity patterns over space and time and across biogeographic realms, both terrestrial and marine, and also across a variety of taxonomy groups. Current research teams in his lab are biodiversity monitoring with cameras and sound recorders, mapping functional diversity of pollinators, employing eDNA for biodiversity modeling, and assessing nature restoration activities using integrated modeling approaches. So as you can see, Dr. Kass's interests and research activities are pretty broad, and we are very excited to hear about just a small fraction of them today. So again, thank you all very much for coming to our Funk lecture. And thank you, Dr. Kass, for being willing to give the seminar talk for us. You can now share your slides and take it off.
Okay, thank you. Give me one moment here.
Sure.
Okay. Okay, can you see my slide?
Yes, it's there. Thank you.
Great.
Looks good.
Okay, thank you so much. Okay, so I'm just going to begin. Okay, so thank you very much for the introduction, Marta. As she said, my name is Jamie Kass. I'm an associate professor at the Graduate School of Life Sciences at Tohoku University, which is in northern Japan, and I run the macroecology lab. I'll be giving a talk today entitled Addressing Wallacean Shortfalls for Understudied Taxa with Species Distribution Models. This is part of this series, the Funk Lecture series on biogeography. I'd like to thank the International Biogeography Society for inviting me to give this talk. And without further ado, I will begin. I can only change this slide. There we go. So just a little bit about me first. So I am a biogeographer and a macroecologist. I have interests in biodiversity modeling and mapping and a few other things. I run this lab with some great assistant professors, Professor Miranda and Professor Takeshige. And we have a lot of students in the lab working on various projects, very interesting projects that span a wide variety of taxa, as you can see. These include mammals, birds, lizards, fish, flowering plants, mites, zooplankton. And this is just what we're studying now. Who knows what we'll study in the future? I often get asked as an American, how did you end up working in Japan? And so I included this map below, which shows my very not straight career path, bouncing back and forth between countries for various fellowships in different positions and schools and degrees and jobs. Unfortunately, going over this would take up all our time. And so unfortunately, I can't go into detail about this, but what I wanted to demonstrate is that it's complicated. So today's talk will be broken up into 5 parts. The first is concerning Wallacean shortfalls for understudied taxa in the age of big data, which is now. The second is, how do we make use of little data that we have for these understudied taxa? Next, I will discuss a few examples, project examples that show how we can make regional and global maps of insect diversity. Insects are one of the major understudied taxa groups. Next, I will discuss how we can use functional diversity maps to make predictions of provisions of ecosystem services, which is particularly important for invertebrates, which are major contributors to ecosystem services. So this part of the talk will focus on, after we have our range estimates, and what can we do with them? What kinds of applications can we do with them to make new information that's important for management and conservation? And then finally, I will end with some brief thoughts on how I think we should be moving forward in the field. So I hardly need to impress upon this group the importance of species distributions. So I decided to dedicate a single slide to this. So without biogeographic information for species, we cannot do the following. We cannot prioritize conservation areas, otherwise known as hotspots sometimes. We cannot make environmental management plans, or we cannot inform them. We also cannot assess invasion risk for alien species. We cannot predict range shifts for species with climate change or other kinds of anthropogenic pressures. It's going to be very hard to test certain ecological theories, like the latitudinal diversity gradient, without biogeographic information, and then many, many more. And so the lack of biogeographic information for species is known as a Wallacean shortfall, after the founder of modern biogeography, Alfred Russel Wallace, who is shown here in very fancy clothes, half sitting on a chair in his younger and fancier days. So we currently live in the age of big biodiversity data. And this means we have relatively comprehensive taxonomic and geographic data for tetrapods in particular. Tetrapods include amphibians, reptiles, birds, and mammals. We also have data on some fish groups, particularly those of commercial value, and also for plant groups, particularly trees. For everything else, very little. And so the maps below are global diversity maps that were made approximately 10 years ago using expert maps. So these are maps created by experts in these different taxonomic groups. And these authors, the researchers combine these maps to make global biodiversity maps for each group. These have been around for quite some time. We were able to do this because we have big databases that host expert map information. And one of the most important ones is the IUCN Red List database shown here that additionally gives different measures of extinction risk for species. However, most of this information is focused on tetrapods. And so here we can see an expert range map and some extinction risk information for the giant salamander in Japan. However, Wallacean shortfalls persist even in this age of big data. As many of us might know, most of animal biomass is invertebrates. So here's a diagram from a recent paper showing all the animal biomass in the world, estimates for animal biomass. And the tetrapods, I've put orange boxes around. What's horrifying is that livestock and humans make up most of the tetrapod biomass, whereas wild mammals and wild birds make up only a sliver. And reptiles and amphibians were so small that they weren't even included in this diagram. The rest of it, the bulk of it, is invertebrates and fish. And mind you, most fish species are understudied. So one of the root problems here are Linnaean shortfalls. These are shortfalls in taxonomic information. So for a lot of these understudied groups, we just haven't described most of the extant species yet. A recent estimate says that about 80% of insects are still waiting to be discovered. This is just insects. You can imagine how many other groups are also waiting to be discovered. How many other groups have large percentages of species that are still waiting to be discovered. However, even described species lack geographic data. Even species that that taxonomists have described for understudied groups typically lack occurrence data and expert maps. And most of these species have close to no data associated with them, no geographic data. Why? Why is this the case? We lack research funds. So here's a diagram showing average funding per taxon in euros for different groups. And you can see here are the tetrapods, here's fish, and here's everything else. Well, there's only 3 groups shown here. And you can imagine what it's like for cnidarians. It's probably all the way down here. So most of it is for tetrapods. We also lack dedicated databases for understudied taxa. And we have a lot of issues with data sharing. So we have a lot of big open databases, but they focus mostly on tetrapods because most of the data is focused on them. Most of the research attention is focused on them. And so thus we have very few global maps for invertebrate diversity. So how can we make use of the little data that we have for these understudied taxa? What can we do? How can we get biodiversity estimates? So I'm going to present a non-exhaustive list of strategies. The first one is to make regional summaries. So these are summaries of existing data, typically occurrence points, sometimes species lists, that are aggregated to a regional scale, typically some kind of administrative boundary, like a country or sub-country scale. The advantages here are that you can use a variety of data types. Also, the scale is relatively coarse, the spatial scale. And so this is good because a lot of this data has high uncertainty associated with it. However, some disadvantages are that the units typically lack ecological meaning. Countries don't have any ecological meaning. Animals and plants happily cross country boundaries all the time. And also there can be very sharp transitions from one polygon to another, which is not very ecologically realistic. And below are some examples of regional summary maps that were made for different invertebrate groups. I'll be showing bees many times for different methods because in this paper they use a lot of different methods. And so if you're interested in comparing them, you can check out this paper by Orr et al. 2021. The next two options are related in the sense that they use estimates per species and then combine those species estimates to make biodiversity maps. So the first one is expert maps. We just talked about those a bit. The advantages here are that they represent the current knowledge of the species, and that because of that, they have less sampling bias than opportunistic occurrence data. The disadvantages are that they have possible spatial biases. So in this paper by Hughes et al., they pointed out that a lot of these expert maps tend to follow administrative boundaries. So in other words, they might have the same disadvantages that the regional summaries do. Convex or alpha holes shown here, which are basically shapes that are drawn to contain all the point data associated with a species, so occurrence data, are often used in place of expert maps when they don't exist. So we find this often used for understudied taxa. And these have the advantage of showing the true limits of the data. So it shows the true spatial extent of all the data that is known. In particular, alpha holes have flexible shapes. You can define a distance threshold that allows the algorithm to make new shapes for point clouds that are quite far from each other. And so there's a lot of flexibility. However, disadvantages are that when the shapes get very big, when the species has a large range, it can include dispersal boundaries. So it can include big rivers or mountain ranges that present dispersal boundaries for the species, and also it can include unsuitable habitats within that range. And so they're not they might be relatively inaccurate if they include a lot of unsuitable habitat inside, or they cross dispersal boundaries. Also, they're limited by available data. And as we know, understudied taxa tend to have big sampling biases, geographic sampling biases. And so because these represent shapes drawn around data points, they're limited by that data. Next, I'll talk about two modeling approaches. The first is macroecological models. These are models that predict biodiversity indices directly using typically environmental predictor variables. And most typically, we're predicting richness. So it's richness predicted with environment. And so here are two examples below, one for bees and one for earthworms that were quite recent. Advantages here are that these models use species inventory data, which are structured, which have less biases than opportunistic occurrence data. This is good. Also, they are based on macroecological theory, particularly that communities have carrying capacities that are defined by environmental variables. And so this can be attractive. Disadvantages are that species inventories themselves are quite scarce. And also, they're not evenly distributed across the globe. They're hard to find. There are exceptions to the rule, but typically, especially for understudied taxa, they're hard to come by. If you have them and you have these data sets, these models can be good to use. Another disadvantage is that macroecological models do not produce predictions per species, and they also don't produce predictions of community composition. And we'll see why that could be important. And then finally, the core of this talk, species distribution models, shown below with some, in these two examples that I will describe a bit later in the talk for this bee genus, Anthophora. So the advantages of species distribution models is that you get predictions per species, And you can also get community composition predictions, and I'll explain how you can get those later. You can also limit your predictions to the extent of the data. So you can make predictions inside holes. You can draw a shape around your points and then make a prediction inside that shape. And I'll show you how that's done in a few slides. Some disadvantages are that species interactions are not considered typically. And so if you don't consider species interactions, it's quite easy to make over predictions of species richness when you combine many models together. And also, these models use opportunistic occurrence data and therefore tend to have quite large sampling biases that then need to be addressed. Very importantly, these modeling approaches have the additional ability that they can predict areas or times with no species data. The other approaches cannot do this, and that's very important. So for those of us who are not well-versed in species distribution models or SDMs, I wanted to show this simple diagram going over what the workflow looks like. And so today I'm going to be talking about species distribution models that use background records instead of absence data. Absence data is hard to find. It also has high uncertainty. And so typically these days when people make SDMs, they use background records. Not in all cases, but in many they do. And so background records refer to a random sample of the available environment for the species. And so you're comparing that random sample of the available environment with the species occurrences, as opposed to comparing presence to absence. You then match these with environmental predictor variables that can include climate, topography, land cover, etc. And then you build a model, which can be a regression model or a machine learning model. Today, I'll be talking mostly about machine learning models. And you make a prediction. The prediction can be an environmental prediction that can show the species response to environmental variables, or it can be a map which shows the species potential distribution, the predicted potential distribution. The nice thing about these models is that although they are made per species, they're made per individual species, you can combine them or stack them to estimate biodiversity metrics, for example, species richness or community composition, to estimate beta diversity, etc. And as mentioned before, if you input new environmental data into the model, you can get predictions for areas or times with no species data. So for example, you can make a prediction for the future to estimate range shifts via climate change, and you can also make predictions of ancestral ranges given paleoclimate data. So a lot of attractive characteristics. I just want to mention that there are many available tools for SDMs. Just last year, I put out a paper with a big group of ecologist R package developers explaining all the available tools in the R language that are all open and free, and how to combine them to implement best practices for SDMs. I was also part of a development team that made this application called Wallace EcoMod. It's an application that runs through an entire SDM workflow that requires no programming at all. So if you're not comfortable with programming, you can also use applications like this. There are other ones too, to run through the workflow and make models yourself. So there are many tools available. So now I want to talk about how we can make regional and global maps of insect diversity. And first, I'm going to start with regional maps. So this is a project I'm currently working on with some collaborators in Egypt. And so we're working on the research objective is to make distribution predictions for this genus of bees called Anthophora. Anthophora is a genus of ground nesting and solitary bees that are understudied relative to social bees, which get much more research attention. However, solitary bees are also very important pollinators, and so it's important that we also get biodiversity maps for them too. We were focusing on the Mediterranean region, and some of these bees even have distributions in the Middle East, Eastern Europe, and up into continental Europe as well, also into North Africa. However, data was very limited for some species with very large gaps in Eastern Europe, the Middle East, and North Africa. We were able to get some data for countries in some countries in Europe, but we got very little or no data for the Middle East and North Africa. For this species restricted to Egypt, we were lucky enough to have field data that supplemented our analysis, but when we checked open databases, there was almost nothing there. And so, what approaches did we use to make models for these species that were missing a lot of data? So we used Maxent models. This is the machine learning technique, a machine learning algorithm that can be tuned to be either very simple or very complex. And so we forced the models to be very simple. We forced them to produce unimodal quadratic responses. So just kind of curvy responses with a single mode for all variables. Because data was low and missing from large regions, and there was high uncertainty associated with this data. And so you can see here that for the species that has a very small range restricted to Egypt, there were only 3 model coefficients. And for this species with a much larger range, there were 5 model coefficients. However, all the responses are relatively simple. This was also really important because one of the other research objectives was making climate change projections. And when we're making climate change projections, we worry about extrapolation. So having simple model response curves is very attractive when you're doing extrapolation, but also it's important to map areas of extrapolation in order to show areas of high uncertainty. So for this small range species, we can see that most of this big extent is covered in red. Red here means that the environmental conditions in these areas are very different from the training data, which is right here, the environmental conditions associated with the training data. So if we're making a climate change projection for this species, we don't want to go much outside of this box. However, for this species, which ranges all over Europe, even though we're missing much data for this region, for the eastern part of its range, when we made our environmental similarity map using the MES algorithm, we can see that there isn't much red, so it's mostly blue, which means that most of these environmental conditions throughout this range are very similar to the training data. And so if we make a climate change projection for this species, we won't require much extrapolation throughout this extent, which is very important information we need. So I'd like to then talk about scaling up to global diversity maps for invertebrates. And so I'm going to focus here on ants. Ants have dominant roles in ecosystems, and therefore they're a good proxy for other invertebrate groups. Ants, so we have a lot of data for some regions for ants, but we have been lacking data traditionally for key parts of the world. So you can see this is an older study that used existing ant data. You can see there are large geographic gaps all over the world, but they did the best they could with the data they had, and they were able to make good point level estimates. This study by Jenkins et al., it used a macroecological modeling approach, like I explained earlier, using data very similar to this to make predictions for the world, but in areas of very high uncertainty that had very little data or required too much extrapolation, they removed these from the analysis, and those are in black. And so more recently, a database came out called Gabi, which is shown in antmaps.org. You can go check it out yourself. At the time of the study I will describe next, there were over 2.5 million records and probably more now. Gabi is a unique database because it was able to accumulate very good sampling for the entire globe. The way it was able to do that was by mining the literature in a range of different languages. So this is in addition to getting data from open databases and also from personal collections of ant scientists. And so it accumulated lots and lots of data together. And in this paper, Economo et al., they were able to make very good estimates for sub-country administrative regions for the globe. However, what we wanted to do was get grid level estimates for ant diversity. And so in order to do that, we would need to use a species distribution modeling approach. So I will now go over the workflow we used to tackle this. So the first one is this data informatics pipeline. It looks a bit complicated. So this first step is how the Gabi database was produced. The next step shows data cleaning, so removing dubious data points, and also geocoding, which is very, very important. So a lot of the data we had 2.5 million points approximately. A lot of these points, a lot of these point records had locality information that was text only, so it didn't include coordinates at all. We need coordinates in order to match with the environmental variables to make our models. And so we used a geocoding pipeline to produce coordinates using Google Map services for these records that were missing the coordinate information. And so this is the before, this is the after. And you can see there's so much better global sampling after we created new coordinates for records that were missing them before. So this is really important. Next is the species distribution modeling pipeline. And so I'm going to break this down into these main steps. So after we have a finalized occurrence data set per species, we then make a hull. We used alpha hulls, this red shape here, and then we buffered it by 30 kilometers to account for areas just outside the range that might be part of, sorry, just outside the data extent that might be part of the species range. We then spatially thinned the data to remove cell duplicates and also to reduce clustering. We then matched the data to bioclimatic variables, and then we tuned models to get optimal complexity for each one, and we made predictions of potential distributions. And so post cleaning the data, we had a total of over 14,000 species and 1.5 million records. So this is relatively big data for ecology. However, about half of these species had data that looks like this. So they had 1 to 4 records associated with them. It's it's impossible to make a model with 1 point, and you don't want to make a model with 2. And so we decided to be very generous, and we made models for species that had 5 or more records. And we had good evidence in the literature that using certain approaches, you can make reasonable models for species with 5 or more records. And so we did that. For the species that had very few records, 1 to 4, we basically just used buffers for species that had 1 to 2, and we used alpha hulls as is, as the range estimate for species with 3 to 4 records. For those with sufficient data or 5 or greater data points, we made species distribution model and predicted the potential distribution inside of that shape. So these predictions did not go outside of the extent of the data that we were given. Additionally, we had to address pretty severe spatial sampling biases in this data set. And also, we did some things to address data size differences. So when we sample, so to sample our background data, instead of doing it randomly, we used a probabilistic approach. So we estimated the occurrence sampling density for ants for the entire globe. This is just the kernel density of all ant occurrence data that we had. So areas that are very red have high density of ant occurrences in our data set. And we probabilistically sampled this to generate background points for each species in order to emulate the same bias that's in our occurrences in order to cancel the bias out. We also use model tuning to find optimal complexity, for parameter combinations in our models. And so we use this package ENM-eval that I developed we use the cross-validation approach to evaluate models, and then selected models that had optimal complexity based on different evaluation metrics. And so simple simple models, we ended up selecting simpler models for species with low data and slightly more complex models for species with more data. So once we had models for each species, then we were able to combine or stack them together to estimate richness. However, richness in and of itself would render small range species invisible. So when you look at a richness map, most of the contributions are from large range species. Small range species tend to be hard to see in these maps. So in order to make them more visible, in order to emphasize them, we calculated what's called range rarity, which is a weighted richness that increases in value when the range size is smaller. And so we made two kinds of maps, richness and range rarity. Very, very important for understudied groups because a lot of species have very small ranges. And the species with small ranges are of greater conservation concern because they tend to have higher extinction risk. We also confirmed how robust our results were to changes in methods by comparing hull-based, so polygon-based approaches with our model-based approach. So we compared our richness and rarity, our richness and rarity estimates using models for half the species with estimates that use just polygons for all species, just to make sure that our results didn't change too much. And the result was they did not. So the models contributed, but they didn't fundamentally change the answer, which is good news. We were then able to compare ant richness and rarity patterns to those of tetrapods. Here it says vertebrates, but it doesn't include fish. And so what we found was, surprisingly, these patterns tended to match in a lot of places in the world, but in some regions, like for example, Mediterranean, there's much higher ant richness and rarity in the Mediterranean than for these combined vertebrate maps. We also found interesting things like for range rarity for ants, the correlation was highest with reptiles, probably because of a higher affinity to arid areas. And so this was very interesting and probably very useful to do for other understudied taxa, if we were able to make more kinds of diversity maps for them. So after we finished this, we showed the maps to some ant experts, and they pointed to certain areas and said, "This is wrong. This is wrong. You know, this is too low." And we said, "Well, why?" And we took a look, and it was because sampling density was very low in certain areas of the world, and so our estimates were lower for richness than were expected by experts. So we had to fix that. So what we did was we built a new model, a random forest model that predicted our diversity estimates, but used this suite of predictors that included this global sampling density, the one that I showed right here. This was one of the predictors in the model. This model obviously predicted our observed patterns very, very well, but why we did this was because if you build a model with a predictor that includes sampling density, you can then tune this up or down to make different scenarios of sampling. And so what we did was we tuned sampling density up all the way to make a scenario where sampling was equal everywhere in the entire world. So areas that were undersampled now had equal sampling density to areas that had a large density of points. And so under this scenario, we then made a new prediction for richness and a new prediction for rarity in order to try to correct our previous estimates. And so what we got are maps that look like this. So areas that are red are, sorry, I forgot to mention. After we made our richness and rarity estimates, we then mapped diversity centers or hotspots by setting a threshold of the top 10% richness and rarity areas. And so these are top 10% areas. So the highest range rarity areas in the entire globe. And red areas dropped out of this top 10% with our new predictions. So once under our equal sampling for the entire globe scenario, red areas dropped out. They're no longer the top 10%. Purple areas remained stable. And then blue areas were new. These were new diversity centers. And so these popped up in areas that had the lowest sampling. And so Southern Andes was one, Atlantic rainforest was another one, Western Africa, Central Africa, much of Southeast Asia, Western Ghats, part of the Himalayas. These areas probably contain species either unknown to science or species that have data stored in museums or other places that are not being shared with the rest of the world. And so if we want to improve global data sets for ants, these are areas where we need to go and take new samples and or create new collaborations to increase data sharing to kind of get them on the map. And so interestingly, these predicted rarity centers actually make the ant diversity patterns more similar to those of tetrapods. But as we're currently missing the data, it's hard to tell if this is the truth or not. And so what we need to do is prioritize these areas for future ant sampling. So in the limited time we have left, I'd like to quickly talk about how we can use functional diversity maps to make predictions of provisions for ecosystem services. So it's been clear for many years that biodiversity has positive relationships with ecosystem functions and services, what I will abbreviate as ES. Here's an old paper from Tillman et al. That shows all the different positive relationships between about the different kinds of biodiversity, not just richness, but also functional diversity and ecosystem function. Also, as we know, invertebrates are primary drivers of ecosystem services and functions. But as we also have poor spatial resolution for their diversity patterns. So if we can make range estimates for these taxa, we can also map ecosystem functions and services. Traditionally, most of this ecosystem service mapping has been done using simple land use lookup tables. So one land use type equals one kind of service. It's a bit oversimplified because species are primarily responsible for these services. And so if we don't include species data in these predictions, they're not very realistic. And so we need to know biodiversity patterns for the species responsible in order to make accurate maps of ecosystem services and functions. And so with collaborators last year, I published a paper that outlines how we can use advances in biodiversity models to improve ecosystem service predictions. And so this is a diagram showing this schematic. So we can use SCM workflows similar to the ones I just mentioned to make biodiversity maps for ecosystem service providers. So here I will give an example for pollination services, in particular bees. Bees are not the only pollinators, but they tend to have the most data. And so we'll talk about bees today. So some SDM approaches were proposed that link predictions, SDM predictions, with ecosystem service potential calculations like this one that focus on distances to existing crops. However, the focus is on services to crops, but if we want to know in general, how high the potential provision of ecosystem services is in each grid cell, we can use pollinator functional diversity as a good proxy. And so here's an example for Japan. So here, citizen science data was utilized to sample bumblebees all across the country, which enabled SDM construction that wasn't previously possible before, because in all the open databases, there was very little bee occurrence data for Japan. And so citizen science data, the citizen science effort increased the data considerably, which was then uploaded to GBIFs. And so if you check now, there's actually a lot of data there. And so what they did was they thresholded their SDM predictions to make binary maps, and then stacked those to get community composition estimates. So they had estimates for which species were in each grid cell. And then they could link those with functional traits that they measured. And so here they measured proboscis length. This is the bee's tongue, which is related to the kinds of flowers it can take nectar from and pollen. And they matched the proboscis length to each species and then calculated things like minimum length, maximum length, and length range to make maps of these functions for the country, which was really neat. And so I ended up collaborating with some of these authors to go one step further and make multivariate functional diversity maps. So maps that consider multiple traits, not just a single one. And to do so, we need to use multivariate methods on trait data sets. And so here we use a hypervolume approach. So it's kind of like a PCA or an ordination style approach. Each of these is a species, and then you plot them in this trait space, and then you draw a hypervolume around the species points, and then you can calculate the volume of that shape. And the volume is your measurement of functional diversity. So each community would have a volume associated with it that can then be mapped to these grid cells. And so these represent species distribution model, stacked species distribution model predictions of communities, and each one gets a functional diversity value mapped to it. And so here's a preliminary result example from a master's student in my lab who's working on this project with us. And so here's predicted species richness for Japan using SDMs. And then this is the predicted functional diversity using these hypervolume values. And so using this approach, we can make two kinds of maps for different kinds of diversity and then compare them. And we can see, this is preliminary, of course, but we can see potentially some interesting biogeographical differences between these different diversity metrics. So moving forward, just some final thoughts. So we still lack biogeographical data for understudied species. However, using pipelines like the one that I described today and other approaches for regional maps that I described today, we can make diversity maps at different scales for different groups, even using data that's incomplete. And so we need to not only build up these databases for understudied species, but also start to make range estimates for them and get preliminary diversity maps that we can then compare to better known groups. And so we can use, I demonstrated today that we can use different approaches for both high and low data species. We can vary our methods based on how much data each species has. We can tailor models to be more simple. So even using models that are capable of very high complexity, we can force them to be simpler when we're modeling species with low data. And we can also make predictions for different sampling scenarios when we know that we're missing data for key regions. Newer techniques like joint SDMs, which are multi-species models, and also integrated SDMs that use varied data sources in order to make large-scale predictions for things that we typically measure on small scales, like abundance. These will definitely play important roles in filling Wallacean shortfalls for understudied taxa. I didn't talk about them today. They're relatively new, past 10 years or so. And I myself would very much like to use them in the future for these purposes. Some people are even doing it now. However, in the end, we are always limited by quality species data. This is always the limitation. And so what we need the most is more data sharing, more field collections, and more taxonomic analyses to document new species and localities. Thank you very much for listening, and I am happy to take questions with the limited time we have left. Thank you.
Thank you so much, Jamie. This was fantastic. If you have a question for Dr. Kass, please put those in the Q&A box, and I will read them off for Jamie. So as we're waiting for people to ask their questions, I have one that you've already started talking about. Like, there is no substitution for actually getting more data, right? Like, we really need the data, and we really need the data from places where we currently don't have any, or we have very limited data. What do you think are some of the techniques that we can harness in the future to speed that process up? Because taxonomists, the number of taxonomists have been declining over the past a couple of decades quite steadily. So we have fewer and fewer skilled people to actually identify museum collections to species.
Right.
So I don't know, what do you think is the future there?
So one thing that we can do now is use this geocoding approach for records that lack coordinates. So if we have records for species that have limited locality information, we can generate coordinates for them using a similar workflow to what I described today. Another thing is, if we lack data on species, we can also make maps of genus diversity and even family diversity for some groups. So we can just make the same kinds of maps, but at a higher taxonomic level, if that's all the data we have. And even then, it can be very informative. Another thing is developing collaborations with researchers who work in the areas where we have the biggest data gaps. This is maybe the most important thing, developing research collaborations in these areas, because a lot of times people are sitting on data that are not being digitized because of lack of funds. And if you can work with people, maybe to help accelerate that process, museum records or field records can be digitized and shared with the rest of the world. And so, yeah, but yeah, you're right that the limitation is always the data itself. But even when we're missing data, there are always approaches we can use to get informative results that might not be exactly the truth, but can nonetheless be useful.
Great, thank you. There is a question that I will read to you now. It's fairly lengthy and has three sections. So I will just read the first one, and then we can take the second one and so on, so that we don't overwhelm you with the entire question. So that's a question from George Amaro. Professor Kass, first of all, congratulations on your work. I believe I have read a large part of your excellent publications. And here's the question. When we talk about simple models with less complexity for transfer or for predictions under future climate conditions, how simple can these models be in general without compromising the quality of the predictions?
Let me tackle that one first. So in general, simpler models can be preferred when we're making predictions under future climate, because when we're extrapolating, you tend to want to finish your response. So if your response is going, if it's going up and it needs to go down, you want to finish it and make it go all the way down, right? If you're making a complex model that like for example a generalized additive model or something that has many different splines and stuff, when you make an extrapolation, the response is always clamped to the most extreme value in your training data, and then there's just a straight line after that. So complex models have a lot of trouble extrapolating. Simple models don't, because they're based on simple equations. And so that can be attractive. However, a linear model can also be dangerous because it just keeps going up or just keeps going. Well, if it's going down, it's maybe not such a big deal, but it also depends on the direction. So simple models can be attractive for those reasons. But it's also important to understand the uncertainty in these predictions and maybe make different predictions for different models at different complexity levels and just kind of see how much your answer changes. If it doesn't change that much, maybe not a big deal. If you have enormous changes, it's something you want to document, right?
Yeah, great. Thank you. And then the second part of this question is, doesn't geocoding occurrences include a degree of uncertainty in the model data? What is the maximum administrative level that should be considered for this?
Yes. So if you read the paper that I talked about today in Science Advances, it is a very detailed section talking about how we handled uncertainty when we did the geocoding. And so it's kind of complex, but definitely there is uncertainty associated. And some points we removed because the uncertainty was too high. And so definitely check out that paper for that reference.
Okay, great. And then the last part of the question is in regards to invasive species. So considering a model for invasive species such as insect pests, naturally with different objectives than a model for plant conservation, using biogeographic entities such as Köppen climate classes to delimit the calibration area considers that these species are not restricted in space and includes anthropogenic dispersal as possibility. I would like to know your thoughts on this. So this was a long comment/question, but basically, I think George is asking about your thoughts on modeling and predicting invasive species spread.
Okay, yeah.
I understand it correctly.
It might take a little while to unpack all of it, but basically the question was about invasive species and calibration area. So what I typically see for invasive species predictions is that we use global data on occurrences for both invasive and native ranges in order to get the best estimate possible of the species niche. Once we have a good estimate of that niche, we can make predictions for other areas in order to evaluate invasion risk, for example. Um, so if you make, but if you limited your calibration area to just the native range, you might be missing some of that niche space that would be occupied by areas that the species had already invaded. And so I'm not sure if I answered the question or not, but I was trying to address it without having to unpack it too much.
Oh, we only have so much time, so
Sorry about that.
yeah. And we have time for one last question from an anonymous attendee. What region in the globe do you think we need to prioritize with macroecological research? What
Ooh,
region do we need to prioritize? Yeah.
I can go back. I should go back to my map on showing the our predictions for the globe and areas of new potential hotspots if we went sampling for ants in these regions. But the answer is multiple places in the globe. It's not a single continent even. It's all over the globe, all around the world. And all of these areas need more sampling. Not just field sampling, but also, as I mentioned, collaborations with people in those areas to fill these data gaps. Because a lot of times we have local scientists collecting data and not having the ability to or not wanting to share. And so collaborations can open that up. And so these are only some areas, and just for ants too, just some areas that need more sampling. They tend to be concentrated in the tropics. So if we're talking about one region of the world, I think tropics is definitely that region, which is quite large. But also this encompasses every continent that has tropical environments in it.
Thank you. We are out of time, sadly, because we would love to hear more from Jamie. If you would like to re-listen to the seminar, we will post the recording on YouTube, on our YouTube channel soon. Thank you very much, Dr. Kass, for joining us again. It was really fascinating, and I think a lot of people really enjoyed this talk, and that's the testament to the, to your research program that we attracted such a wide audience today. So thank you very much again. And for everyone in the audience, we again will be hosting Dr. Mario Moura from Brazil for our next seminar series in July. So please tune in for that, and we will be announcing it soon on Blue Sky and on our website as well. So thank you all very much for joining again. Thank you, Dr. Kass, and till next time.
Thank
Bye,
you very
everyone.
much for having me, and thank you everyone for listening.
Bye.
Bye-bye.